The nephron in the kidney helps remove excess salt from our blood, even when we eat a little bit too much of it. Today's lesson, we're going to take an in-depth look at the nephron, the functional unit of the kidney. So each nephron is a small, independent functioning filtering unit, and there's over 1 million nephrons in each kidney. It allows blood to be filtered out for components to be moved from the filtrate back into the blood, from the blood back into the filtrate to maintain homeostatic balance in our system. So nephrons filter water and solutes from the blood, so things that have been dissolved in the water. Most of the filtrate is then going to be reabsorbed and returned to the blood because most of it is water. In the end, an amber color liquid known as urine enters the tube-like collecting ducts, which is going to go on to the central kidney cavity and go from our ureter to our bladder. Each nephron has its own unique blood supply and its own collecting tubule, which leads to the ureter. Blood is going to enter the nephron through the afferent arteriole, a small branch of the renal artery. So the renal artery is going to branch off into different arterioles, and each nephron has its own. Impurities are then going to be filtered out and emptied into the collecting tubule. The purified blood is then going to leave the nephron through the efferent arteriole. So here we see the structure of the nephron within the kidney. So we have our renal cortex, which contains our Bowman's capsule and Nephron tubule, and our renal medulla, which contains the collecting ducts. So when we look at our nephron anatomy, we have a glomerulus, which is a microscopic glob of capillaries with greater than normal pressure because of the size discrepancy between the afferent and efferent arterioles. So there's a lot of pressure in the arteries in the glomerulus, which forces impurities out of the plasma and into the Bowman's capsule, which is a cup-like structure that collects the leaking fluid forced out of the glomerulus and sends it into the proximal convoluted tubule. So the glomerulus is a kind of blob of capillaries contained within Bowman's capsule. The proximal convoluted tubule is a thin, hollow tube where most of the water and many dissolved particles are reabsorbed back into the blood, both by diffusion and active transport. This is followed by the loop of Henle, which drops down and then back up, actively pumps sodium chloride out of the loop of Henle, creating a hypertoxic extracellular fluid. So water will then be drawn out through osmosis out of the loop of Henle and the collecting duct. So we actively pump out sodium chloride from the loop of Henle so that water can be drawn out with osmosis. The distal convoluted tubule is where various toxins, waste products, and even some drugs can be moved via active transport from the blood into the nephron. So this is where our body uses active transport to move compounds that didn't just get filtered out from the blood into the distal convoluted tubule using active transport, so using energy. Then the collecting duct is where the fluid that is going to constitute urine gets collected. And inside this tubule, most of the last of the water is going to be reabsorbed back into the bloodstream. The remaining urine is going to empty into the renal pelvis and into the urinary bladder. So here again, we're going to look at the nephron structure. So at the very top, we have the glomerulus contained in Bowman's capsule. And then we have the afferent and efferent arteries. These are all going to be contained in the renal cortex, or the outer part of the kidney. Again, the glomerulus, there's a high pressure in the capillaries there, forcing fluid out of the blood that will be collected in Bowman's capsule. So the process of blood purification involves three separate processes. We have the glomerular filtration, tubular reabsorption, and tubular secretion. So in glomerular filtration, blood enters the nephron, flowing through the glomerulus, which is encased in Bowman's capsule. So there we can see the afferent arteriole coming in from the bottom, the efferent arteriole leaving the top, and the glomerulus in kind of a purple color contained within Bowman's capsule. And because of the pressure, substances are forced out of the blood and collected in Bowman's capsule, which will then leave into the proximal tubule on the far right-hand side. The blood is under pressure and the walls of the capillaries and Bowman's capsule are permeable. So the fluid from the blood filters into Bowman's capsule. Now the material filtered from the blood is going to flow through a portion of the nephron called the renal tubule, which is a long tube with semi-permeable walls. The renal tubule consists of the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. So we can see the proximal convoluted tubule right after the glomerulus capillary network in this diagram. The loop of Henle then drops down and comes back up, and then the distal convoluted tubule is on the right-hand side of the picture just before the collecting duct. The materials that are filtered from the blood are known as the filtrate. It contains water, urea, glucose, salts, amino acids, and vitamins. Typically, plasma proteins, cells, and platelets are too large to pass through the membrane of the Bowman's capsule, so those remain in the blood. Now, in the proximal tubule, most of the materials removed from the blood make their way back into the blood, known as during a process known as tubular reabsorption. Roughly 2,000 liters of blood pass through the kidneys every day, and about 180 liters of filtrate pass from the blood into the collecting tubules every day. Now, if we excreted all of this, we'd have to drink about a liter of water every 10 minutes to maintain a water balance. But luckily, about 99% of the water that's filtered into Bowman's capsule will be reabsorbed back into the blood. So our second process is known as tubular reabsorption. Now, this proceeds along the tubular parts by both passive and active transport. So glucose and sodium are removed from the filtrate back into the blood via active transport. And then chlorine and bicarbonate follow those positively charged ions due to a charge. So they are now going to follow with passive transport because of an electrical charge difference. Most reabsorption occurs in the proximal tubule. About 75% of the water in the filtrate returns to the capillaries by osmosis, which is again passive transport because we've increased the concentration gradient of the blood by reabsorbing the glucose, sodium, chlorine, and bicarbonate ions. So because the sodium, glucose, and other solutes are moved via active transport from the tubule back into the blood, water and negatively charged ions can follow by osmosis and passive transport. So again, one more diagram. We move those positively charged ions through active transport. Negatively charged ions are attracted to them, so they follow through passive transport as well. Because of the increased solute concentration in the blood, water flows through from the filtrate to the blood via osmosis. Now, when we're looking at the loop of Henle, the main function of the loop of Henle is to reabsorb water and ions from the glomerular filtrate back into the blood. So as it descends into the renal medulla, the medulla region of the kidney, it encounters an increasingly salty environment, which allows it to become water to move over by osmosis. So because the walls of the descending loop of Henle are permeable to water, but much less permeable to salt, the salty environment of the medulla is going to draw out water by osmosis back into the blood. Now, the filtrate at the bottom of the loop of Henle is now at its maximum solute concentration, because remember, during the descending loop of Henle, the water was able to permeate out of the filtrate, but salt was not. So now we have a very concentrated solute at the bottom of the loop of Henle. Now, as the filtrate continues up the ascending loop of Henle, the walls of the tubule change in structure, and they become impermeable to water and more permeable to solutes. So this picture, on our way down, we see on the right-hand side, water is being taken out of the descending loop of Henle via osmosis and passive transport. And then in the ascending loop of Henle, we see sodium and chlorine taken out through both passive and active transport. We also have a concentration gradient in the center of the tube. So we see that at the top of the descending loop of Henle, we have a concentration of about 300. At the bottom of the loop of Henle, roughly 1200, where it becomes more dilute again near the top of the ascending loop of Henle at 300 and 100. Now, before the collecting duct, we have the distal tubule, which is where material that remains in the filtrate is now still excess salts, water, and urea that our body doesn't need to absorb. At this point, we can have active reabsorption of sodium ions, again, positive, and then passive reabsorption of bicarbonate and chlorine ions, and water is going to continue. But this depends on the need of the body and is regulated by hormones. When the filtrate reaches the distal convoluted tubule, some additional substances pass from the blood into the filtrate through a process called secretion. Now, these substances include nitrogenous wastes, hydrogen ions, potassium ions, some drugs, and other toxic materials. And the pH of the blood is adjusted by the hydrogen ions that are secreted from the blood into the filtrate. So this is another way that the body can maintain its optimal blood pH. What's left of the filtrate passes from the distal convoluted tubule into the collecting duct of the nephron. There is still a lot of water in this filtrate, and as the collecting tubule descends through the medulla of the kidney where the surrounding tissue is salty, more water is drawn out of the filtrate, concentrating the urine further. So the reabsorption of water causes the filtrate to become about 4 times more concentrated from the top of the collecting duct to the bottom. And now only about 1% of the original filtrate from Bowman's capsule remains, and it's now called urine. So in Bowman's capsule, we have a secretion of fluid from the blood. In the proximal tubule, we have active reabsorption of positive ions and passive reabsorption of negative ions, as well as water. In the descending loop of Henle, water is passively reabsorbed because of an increasingly salty environment. In the ascending loop of Henle, we have the active transport of positive ions and the passive transport of negative ions. In the distal convoluted tubule, again, we have the passive transport of negative ions following the active transport of positive ions, as well as the tubular secretion using active transport of other substances in the blood that need to be removed. Now, as the filtrate travels down the collecting duct, water is again reabsorbed until urine is formed and passed from the ureter into the bladder. So urine from the millions of collecting ducts are now going to travel through the renal pelvis into the ureter, which is going to lead into the bladder until it can be released through the urethra. Now, the urethra in a female is much shorter than the urethra in a male, leading them to more have a higher risk of urinary tract infections. Okay, well, that is our lesson on the structure of and function of the nephron in the kidney. Thanks for joining me. I'll see you again next time. Bye.